in the second day of 5 days short-term training program on environmental impacts of COVID-19 pandemic, challenges and remedies through science and engineering. Let's begin with the day 2, session 1 on the topic environmental oxygen and their impacts on sustainability to obesity by Dr. Nighat Fahmi. Dr. Nighat Fahmi, Associate Professor, Deputy Director, APTC, University of Rajasthan, Jaipur. She has completed her PhD from University of Rajasthan. Her major areas of research interest are synthetic inorganic, organometallics, macrocyclic, green and bioinorganic chemistry. Dr. Nighat has awarded a certificate of appreciation for research work by University of Rajasthan. She was Vice Principal of University Maharaja College in 2010 and 11. She is NSS Program Officer in 2011 to 2013, coordinator in the UGC-sponsored refresher course Eco-Friendly Benign Chemistry in 28 November 2011 to 17 December 2011. Resource person for refresher courses being organized by ASC University of Rajasthan, Jaipur. Her total teaching experience is more than 21 years. She has successfully completed 2 UGC major and minor projects, reviewed papers on the reputed international journals, life member of various academic societies. Dr. Nighat has supervised 70 PhD students and has more than 85 research publications in international journals of repute. She published books on applied chemistry for engineers, Paragon International Publishers, New Delhi. She has attended 30 conferences, seminars, workshops, and recently 6 PhD students are working under her guidance. Please, ma'am.
Oh, yeah. Yeah, thank you very much, Dr. Anurika, for your kind introduction. Let me at the outset thanks Professor Dinesh Goyal, Principal Director of Poornima Institute of Engineering and Technology, Dr. Sama Jain, HOD Applied Sciences, Dr. Gautam Singh, Convener and Registrar, and Dr. Manoj Bajpai, Convener, Poornima Institute of Engineering and Technology, for giving me this opportunity. It is really a privilege and pleasure to be part of this short-term program of such prestigious institutions. And congratulations to organizing committee for arranging such wonderful program. And I'm sure participants and students will definitely benefit from it. And as you know, my broad area of research is synthetic coordination chemistry, bioinorganic and green chemistry. We generally synthesize various coordination compounds derived from transition metal as well as main group metals, and we just synthesize them through greener and sustainable approach. And then we screen them for various biological properties to assess their biological potential. Now, but today's my topic of lecture is environmental obesogens and their impact on susceptibility to obesity. Actually, in 2019 December in Delhi University, I attended a workshop on green chemistry for societal needs. And there, one participant from the UK presented work on obesogens on this topic. And I just want to share this topic with you all, which is very interesting and upcoming area of research. And I will just, and at the end of the lecture, you will be able to relate my topic with your topic of the program and the COVID-19 situation. So I will just give you a brief introduction of what are obesogens, their background, history, and their implication to environment and human health causing obesity and various metabolic disorders. So we'll start with this topic. Yeah, this is environmental obesogens and their impact on susceptibility to obesity. As the name suggests, obesogens are nothing but they are environmental pollutants which cause obesity. Now, as we all know, obesity is a global pandemic in adults as well as in children. Now, the incidence of obesity has reached an all-time high, and this increase is observed worldwide. And there is growing need to understand what are the factors that contribute to obesity and how we can treat it and prevent its associated comorbidities. Now, the predominant medical explanation continues to be that obesity is the result of simple imbalance between excessive calorie intake and insufficient energy expenditure. However, recent studies have demonstrated that simple paradigm, that is the simple paradigm energy expenditure and energy intake, cannot explain the energy increase in BMI, which is seen in recent years. So, and as we all know that genetics is widely believed to be associated with obesity. We say about 30 to 70% of the obesity is heritable. But however, known gene variants can explain only 2.7% of the individual variation in BMI. So, therefore, the two most commonly given explanations, that is genetics and energy balance, cannot fully explain the substantial increase in obesity observed worldwide. So, among the obesogenic environmental factors, a growing body of evidence suggests that the exposure to certain environmental pollutants called obesogens can contribute to the pandemics of obesity, also independently of unhealthy diet and physical activity. We know that there are multiple environmental factors which can affect obesity susceptibility, that is beside diet and exercise. Increasing evidence has shown that environmental pollutants impact human health, and even environmental stresses experienced during fetal development have significant impacts on obesity susceptibility later in life. So we will see how it came into existence. That is obesogen hypothesis. Now, the obesogen hypothesis proposes that there are chemicals in our environment termed obesogens that can affect individual susceptibility to obesity and thus help explain the recent large increase in obesity worldwide. Now, in 2003, Jerry Hindle, he is a scientific program administrator at the National Institute of Environmental Health Sciences. He put endocrine disruptor chemicals and obesity on the same map for the first time. And his idea followed from a proposal originally made by Paula Bailey Hamilton, who was the first to suggest that obesity pandemic is associated with the environmental pollution from chemicals. Now, Hindle's proposal that EDC might be influencing obesity was reasonable because nearly every aspect of the control of appetite, satiety, metabolism, and fat storage is regulated by the endocrine system. Hindle's proposal eventually ignited research in the area of obesity among researchers who are already working on endocrine disruptor chemicals. But the idea of EDC as factor causing obesity was not accepted until it was recognized that EDCs could activate nuclear hormone receptors, which are important for the development of wide adipocytes such as peroxisome proliferated activated receptor gamma and the 9-cis retinoid X receptor, that is RXR. And the identification of chemicals that activated PPAR gamma to promote adipocyte differentiation and wide adipose tissue fat accumulation led to the coining of the term obesogen. So hence the obesogen hypothesis indicates that the correlation between increasing in the incidence of obesity and enhanced industrial chemical production was not at all coincidental, but they are potentially related to each other. Now, how the name obesogen came into existence. That is, what are obesogens? Now we all know that obesogens are environmental pollutants which cause weight gain. Now in early 2000, Bruce Blomberg and Felix Groen of the University of California, Irvine, coined a new term for environmental chemicals linked with fat gains as obesogens. The coining of term obesogens caught the attention of the people, and lots of research came out on these new field of research. Now, how we define obesogens? So, obesogens were defined functionally as chemicals that lead to fat accumulation or that causes weight gain. Obesogens, they are a category of endocrine disruptors chemicals that can interfere with hormones. And some endocrine disruptor exert their effect by activating estrogen receptors, which can cause harmful effects in both women and men. Some obesogens have not only been linked to obesity, but they also cause various metabolic disorders like birth defects, premature puberty in girls, demasculinization in men, breast cancer, and other various disorders. Now, uh, these obesogen chemicals, they may also influence an individual weight and metabolism, even if exposure occurred much earlier in life. That is, unfortunately, many of these effects happen in the womb. For example, when pregnant women are exposed to these chemicals, their child's risk of becoming obese later in life may increase. For example, if a mother smokes during pregnancy, her child has a higher risk of being overweight or obese during childhood and adulthood. Hence, widespread exposure to EDC or obesogen is a likely contributor to the obesity epidemic. Now, which chemicals are obesogenics? Now we know that obesogens are nothing, they are but a class of endocrine disrupting chemicals, and they are present in the environment and named after their potential to induce obesity in mammals. Now, there are many chemicals, more than 52 chemicals have been identified as potential obesogens, and studies of both animals and humans have pointed out to many chemicals as potential obesogens. But some of these chemicals, such as organotins, bisphenol A, and phthalates, with very diverse chemical structure, they were not initially characterized as obesogens, but they were initially characterized as androgenic or estrogenic due to their ability to mimic or alter the uh, role of these reproductive hormones, leading to reproductive dysfunction. But however, recent evidence suggests that these compounds, they also interfere with nuclear receptors that play a key role in adipogenesis or in weight gain. So there is a list of chemicals which are obesogens, and the mechanism of action is also given. And some of these are some of the obesogens on which most of the research work has been performed. So the first is bisphenol A, which is 2,2-bis-4-hydroxyphenyl propane, and it is used in how it came into the environment is because it is used in the production of polycarbonate, plastics, and epoxy resins. And how it acts as a obesogen is that it is an active agonist of estrogen sensitive membrane receptor, that is GPR30, and glucocorticoid receptors, and it mimics the structure and function of hormones, that is estrogens, and it alters glucose metabolism, impairs adipogenesis, and causes adipocyte dysfunction. And next is our phthalates. They are the diester of 1,2-benzene dicarboxylic acid, and they are used as plasticizers. Now they also belong to class of obesogens, and they have anti-androgenic and weakly estrogenic properties, and they influences obesity through mechanisms such as anti-thyroid hormone activities and or by activation of peroxisome proliferator activated receptors, that is PPAR gamma, and epigenetic modulation. It was found that maternal phthalate exposure is associated with alterations in methylation of critical placental genes. Next, obesogen atrazine, and it is the 2-chloro-4-ethylamino 6-isopropylamino 1,3,5-triazine. It is generally used as herbicide, and it is largely used in US. So it is a chemical with androgenic inhibiting and weak estrogenic effects. It binds to complex 1 and 3 of the mitochondrial electron transport system and inhibits the oxidative phosphorylation of mitochondria. It acts as an endocrine disruptor by inhibiting cyclic AMP-specific phosphodiesterase. And organotins, the most commonly the organotins are, which are obesogens, are they are tributyltin and triphenyltin. They are generally used as polyvinyl chloride stabilizer, biocides, and they are mostly used in as antifouling agents in paints for ships. And these chemicals interact with transcriptional regulators such as nuclear and steroid receptors, and they affect nuclear receptor signaling pathway such as retinoid X receptor, PPAR gamma signaling pathway, leading to an alteration of glucose transporter and pro-inflammatory cytokines and lipid and carbohydrate metabolism. And organophosphates. We all know that organophosphates, they are used as insecticides, ophthalmic agents, herbicides, and they are also used in industrial chemicals. And it is a chemical that disrupts and monosodium glutamate. We all know that monosodium glutamate is used as a seasoning to make bland, nutritious foods taste good. It is also found naturally in algae, mushrooms, tomatoes, grapes, and frozen foods, potato chips, salty snacks, sauces, and sausages. So it also impairs the secretion of the gut hormone, glucagon-like peptide, that is GLP-1, which is involved in satiety responses and insulin release. It induces neuronal necrosis in several brain regions, including the hypothalamus. Next is clozapine. It is a dibenzodiazepine derivative. We all know that it is used as medicine in treating schizophrenia-related symptoms. It is also used as antidepressant, and it also functions by altering the function of key metabolic enzymes and affects electron transport chain during oxidative phosphorylation in the mitochondria. It also blocks muscarinic M1 and M2 receptors and inhibits GLP-1 secretion, and it also enhances the production of cytokines that modulate immunological responses, and it promotes inflammation. Then we have polychlorinated biphenyls, which also act as obesogen. They are used as plasticizers in paints, plastics and rubber products, in pigments, dyes, carbonless copy, paper, and in electrical heat transfer and hydraulic equipments. These chemicals are hormonally active substances mimicking the action of the thyroid hormone and estrogen. They disrupt the release of neurotransmitter that regulate neuroendocrine function. They causes alteration in intracellular calcium signaling and affect dopamine release. Then we have organobromines. They are used as flame retardants. It is a chemical that affects androgen, estrogen, sex and thyroid hormone pathways. It interferes with thyroid function and testosterone metabolism. It also increases glycolysis and reduces glucose oxidations. And then we have perfluorooctanoic acid. As we all know that it is used in non-stick cookware and waterproof clothing and stain repellent on carpets, mattresses, and in microwave popcorn bags and other food items. Due to its structural resemblance to fatty acids, it has been found to alter energy metabolism and thyroid hormone homeostasis through the activation of PPAR gamma. Then we have genistein, which is the isoflavone, and it is used as an angiogenesis inhibitor, and as a phytoestrogen. It also alters the expression of metabolic and adipogenic regulators such as PPR gamma. It disrupts the epigenetic regulation, a key adipogenic gene. It also alters expression of lipid metabolism genes, disrupts lipolysis and lipogenesis, and it alters ATP synthesis. And lastly, we have heavy metals like arsenic. It also acts as obesogen, and they are generally used in plastics, mobile phones, solar panels, antiseptics, etc. There are many heavy metals which are known to function as obesogens. The chemicals have been shown to cause diabetes through impairment of glucose-stimulated insulin secretion in pancreatic beta cell, stimulation of pancreatic oxidative damage, and insulin resistance in skeletal muscle, increment of gluconeogenesis in liver, and modulation of other hepatic insulin signaling pathways. Looking at the obesogen and their mechanism of action, we know that these obesogens, they can be grouped under endocrine disruptor chemicals because their mechanism of action involves hormones. All the chemicals act by inhibiting some hormone or the other. Now, many chemicals known to be obesogenic in animals models, they are also associated with increased obesity, prevalence, BMI, and body weight in human also. So, research in this area has increased tremendously, and it was subsequently recognized that obesogens may have more diverse effects on metabolism than just contributing to obesity. Although obesity may be a key contributor to such effects, these include type 2 diabetes, non-alcoholic fatty liver disease, kidney disease, cardiovascular disease, and many more metabolic disorders. Thus, we can say that most obesogens are a subset of large class of chemicals termed metabolic disrupting chemicals, but not all of which are obesogens. So obesogens belong to endocrine disruptor class of chemicals. One more is this is DDT. DDT it is also used as insecticide, and it is banned in some places, and it is also a chemical that impairs the function of visceral adipose tissue and decreases the response to energy surplus, causes neuroendocrine disruption of the reproductive axis. Nicotine, we know that it is found in the tobacco plant, and it is used as medicines and stimulant. It also modulates the action of AMP activated protein kinase, which integrates hormonal and nutritive signals in peripheral organs and hypothalamus, thereby playing a major role in regulation of energy balance, suppresses appetite by activating melanocortin 4 receptors expressed on hypothalamic uh, uh, pro-opium-norcortin neurons. Now, so we know that these obesogens they are nothing but they are endocrine disruptor chemicals. Now, mechanism of action, how these obesogen acts. So generally, obesogen cause weight gain by altering lipid homeostasis to promote adipogenesis and lipid accumulation, and this may occur by multiple mechanisms. Like for example, they may increase the number of fat cells, that is adipocytes. They may act by increasing the size of fat cells, that is adipocytes, storage of fat per cell, or both. They may also act by altering endocrine pathways responsible for control of adipose tissue development. They may also alter hormones that regulate appetite, satiety, and food preferences. They may also alter basal metabolic rate and energy balance to favor storage of calories, altering insulin sensitivity and lipid metabolism in endocrine tissues such as pancreas, adipose tissue, liver, brain, and muscle, etc. So obesogens can act to increase adipocyte number and volume by interfering with transcriptional regulators that control lipid flux, adipocyte proliferation, and adipocyte differentiation, particularly through the peroxisome proliferator activated receptor, that is PPAR, alpha, delta, and gamma. Now, how do obesogens act? Although more than 50 obesogens or potential obesogens have been identified, but there is little mechanistic understanding of how most function, because this field is only 15 years old. So we know most about tributol because lots of work has been performed on TBT and its mechanism is documented by various researchers. So like we can take the example of TBT as a prototype and explain how these obesogens work and how they increase weight gain in animals and humans. So TBT is a nanomolar affinity ligand that activates the so-called master regulator of adipogenesis. That is, PPR gamma is the master regulator of adipogenesis, and its heterodimeric partner, that is 9-cis retinoic acid receptor RXR, to promote adipogenesis and alter lipid homeostasis. So we'll learn about TBT first. So this is tributyltin. It is an organometallic xenobiotic that belongs to the organotin family of compounds. And organotins are synthetic chemical. They are tetravalent derivatives of tin4 with general formula, and where R represent organic substituents and X might be a halide, anion, or an organic group linked covalently through a heteroatom, that is oxygen, nitrogen, sulfur, or chlorine. So the compound is prepared by a simple redistribution reaction by combining stannic chloride and tetrabutyl tin. Now, generally, tributyl tin chloride hydrolyzes to its oxide, and it is used as antifouling agents in various paints for painting ships. Now, tributyl tin chloride, it is also used as a precursor to other organotin compounds and reagents such as tributyl tin hydride, and it is a colorless liquid that is soluble in organic solvents. The molecule has tetrahedral geometry and it is widely used in industry. TBT is widely used in industry, and triphenyl tin, it is used in agriculture. And human exposure occurs through dietary sources from contaminated seafood and shellfish, because TBT is used as antifouling agent in paints, and from organotin use as fungicides and miticides on food crops. So through food crop we can also ingest TBT, and in food treatments and in industrial water systems is also the source of exposure to TBT. And TBT contaminates plastics, that is polyvinyl chloride, and it is also present in the house dust. Now, at the molecular level, organotins perturb biological systems via multiple mechanisms reflecting their distinct chemical and physical properties. Now, their biological activity is strongly dependent on the number and nature of the organic groups. So up to a certain length, if you increase the nature of chain length of this organic group, the activity increases, and at a certain point, it again decreases. So it depends upon the nature of the organic groups. Now, mechanism of TBT. Now, as we all know that it targets nuclear hormone receptors. Now, receptor ligands, they include wide variety of small lipophilic hormone and signaling molecules, including classical steroids such as estrogens, androgens, and glucocorticoids, and non-steroidal hormones and dietary vitamins, lipid metabolites, and xenobiotics such as EDCs. So TBT can target nuclear receptor signaling pathway by virtue of mimicking these endogenous ligands, and the resultant unregulated activation of nuclear receptor gene networks leads to inappropriate non-physiological responses. So now Lee et al. Have shown the 3D crystal structure of this TBT and have shown that TBT act as agonist ligand of RXRS, and it has been confirmed by the 3D crystal structure of TBT bound to RXR alpha ligand binding domain via thio and tin bond. So this is the crystal structure of TBT, uh, which is acting as an agonist ligand of RXR alpha. Now, in picture A shows the 3D crystal structure of RX alpha ligand binding domain bound to its agonist ligand 9-cis retinoic acid, and B show the 3D crystal structure of TBT bound to RX alpha ligand binding domain. So here Lee et al. Definitely confirmed the result in their crystal structure models of TBT. So hence it can be proved that the TBT acts as agonist ligand of RXR. No, MSC lineage, this is mesenchymal stem cell lineages. Now, adipose mesenchymal cells, they give rise to various different types of cells, that is osteocytes. You can see here osteocytes on the left side of the diagram, then you have chondrocytes on the right side of the diagram, and here you have myocytes and adipocyte lineages. Now, you can see white adipocyte and brown adipocyte. Now, fat tissue, they can be divided into two principal types: white adipocyte, adipose fat tissue, that is VAT, and brown adipose tissue, that is BAT, based on developmental lineage, morphology, and function. Both lineages originate from multipotent mesenchymal stem cells capable of generating adipocytes, myocytes, osteocytes, and chondrocytes. Now, when we see at the diagram here, we see that adipogenic signals act on committed white pre-adipocytes to induce differentiation into mature white adipocytes. Here, these are the committed white pre-adipocytes, which lead to differentiation into white adipocytes, and white adipocytes will be having adiponectin in it, which controls the lipid accumulation and adipogenesis. Now, when TBT is exposed to human or humans and mouse mesenchymal cells, pre-adipocytes were induced to differentiate into adipocytes via PPR gamma dependent pathway after exposure to nanomolar level of TBT, and exposure diverted bone marrow derived mesenchymal cells preferentially towards the adipose lineage and away from the bone lineage in the exposed mice. Now, suppose we induced exposure to TBT, it acts by RX PPR dependent pathway, and it leads to formation of white adipocyte instead of bone, that is osteocytes. And here brown adipocytes, they are generated from myogenic precursor, and they are responsible for thermogenesis. But here we see that the TBT mimic the effects of fast food that we consume. So what we eat, it increases the fat deposition or lipid accumulation. So also TBT act by PRX, PPR, gamma dependent pathway. It stops the formation bones. Instead, it differentiate into white adipocytes, that is fat. So we can say that exposure to TBT produce mesenchymal cells that differentiated preferentially into adipocytes rather than bone. So this is the uh, mechanism of TBT. And this is schema of the obesogenic effects of TBT on reproductive and metabolic function in adult female rats. Now here we can see in the picture control, that is the rats which are not treated with TBT, and on the right side of the diagram we see TBT exposure of TB to the female rats. So now here we can see there is normal secretion of gonadotropin, there is normal secretion of reproductive hormones, and the ovaries are normal. Secretion of estrogen is normal, and uterus seems to be normal, which lead to normal ovulation and normal formation of corpus luteum, which results in normal reproductive tract functions and leading to normal fertility in adult female rats. And here we can see that metabolic function is also normal. That is, white fat is normal, liver is normal, pancreas and heart is all normal with normal cardiometabolic functions. So control have all the functions, that is reproductive as well as metabolic functions, normal. Now when we look at the uh, picture where female rats are exposed to TBT, we see that there is abnormal secretion of gonadotropin, and there is decrease in the secretion of reproductive hormones, and there is decrease in secretion of estrogen and increase in secretion of testosterone, so which leads to abnormal ovulation, and there is decrease formation of corpus luteum, which leads to cystic follicles and ending up into abnormal reproductive tract functions, leading to abnormal fertility. And here metabolic function, that is in white adipose tissue, there is lipid accumulation by PPAR gamma dependent pathway, and the level of leptin increases. Leptin is a hormone. It helps to regulate energy balance by inhibiting hunger, which in turn diminishes fat storage in adipose site. And there is decrease in level of adiponectin, which regulates lipid and glucose metabolism. It also influences the body's response to insulin. High levels of adiponectin associated with a reduced risk of heart attack. And there is lipid accumulation by PPR gamma dependent pathway in liver. You can see yellow droplets, and there is insulin resistance, and there is abnormal cardiovascular function also due to the exposure of TBT. And PCPOS, that is TBT, induces reproductive abnormalities associated with polycystic ovary syndrome, which is one of the most common endocrine disorders among reproductive age women. So uh, when female rats, they are exposed to TBT, they cause abnormal reproductive as well as abnormal metabolic function in female rats. So hence, it can be proved that TBT acts as the obesogen by disturbing various metabolic and reproductive function of female rats. Now, obesogenic chemicals that may be present in your home at very this moment. So these are the common obesogens which we encounter in day-to-day life. So first one is bisphenol A. It is a synthetic compound, as we know, and it is found in many types of products, that is plastic, food and beverages containers, and baby bottles, as well as metal food cans, etc. And it has been in commercial use for many decades. But recent studies have shown that high levels may cause harm to both animals and humans. BPA structure resembles estradiol, which is the most important form of the female sex hormone, estrogen. As a result, BPA binds to estrogen receptors inside the body. Now, it appears that at the time of greatest sensitivity to BPA is in the womb. Interestingly, 96% of pregnant women in US test positive for BPA in their urine. So BPA exposure has also been linked to insulin resistance, heart disease, diabetes, neurological disorders, thyroid dysfunction, cancer, genital malformation, and many more. While all scientists agree that BPA causes harm at high levels, but there is still some debate over whether it is harmful at the low levels found in foods. Now, next one is phthalates. They are the chemicals used to make plastic soft and flexible. They are found in various products, including food containers, toys, beauty products, pharmaceuticals, shower curtains, and paint. These chemicals can easily leach out of plastics and contaminate foods, the water supply, and even the very air we breathe. So phthalates may be contributing to increased susceptibility to weight gain by affecting hormone receptors called PPAR gamma, which are involved in metabolism. Studies in humans have shown that phthalate levels in the body are associated with obesity, increased waist circumference, and insulin resistance. It appears that men are particularly susceptible to phthalates. And studies show that phthalate exposure in the womb leads to genital malformation and low testosterone level. And phthalate metabolites, they are also found in blood, which is correlated with the type 2 diabetes. Atrazine is one of the most widely used herbicide in US. It has been banned in Europe for over a decade because of groundwater contamination. Atrazine also an endocrine disruptor, and several studies show that exposure correlates with birth defects in humans. In US, there is an overlap between the areas that use the most atrazine and the prevalence of obesity. So several studies have associated atrazine exposure with an increased obesity risk, and high levels may promote weight gain. Now we know organotins. We have read about organotins, and TBT is the most studied one, and it is used as a fungicide and applied to boats and ships to prevent the growth of marine organisms on the hull. It is also used in wood preservatives and some industrial water. And next one is perfluorooctanoic acid. It is a synthetic compound uh, used for various purposes, and it is a constituent of non-stick cookware made with Teflon, and it is also found in microwave popcorn bags, furniture, and food containers also. It has been found in blood of more than 98% of the Americans. It has been associated with various diseases in humans, including thyroid disorders, low birth weight, and chronic kidney disease. Now, exposure to perfluorooctanoic acid during development led to increased insulin, leptin, and body weight during midlife. So, uh, it is also associated with various diseases in humans, and it shows that prenatal exposure leads to weight gain uh, in midlife. So, and the next one is organophosphates. As we all know, previously organophosphate insecticides had been associated with neuropsychological conditions, but researchers found that low levels of organophosphate insecticides had weight gain, which is reported in comparison to their controls. Now, DDT, neonicotinoids, and organochloride pesticides are all examples of pesticides that behave as obesogens. And the ability to accumulate in human fat tissue enables a disruption in the endocrine system, increasing sensitivity to metabolic disorders. Now, pesticides have variety of effects on endocrine system, some affecting the number of fat cells, size of fat cells, or disrupting hormones involved in appetite and food preferences and energy metabolism. So they also lead to obesity and type 2 diabetes. And pesticides and their byproducts are obesogens, and they increases the sensitivity to obesity, type 2 diabetes, and other metabolic disorders, which calls for the systematic changes in pesticide usage and their regulation. Now, how to minimize your exposure to obesogens? So we can avoid food and beverages that have been stored in plastic containers. We can use quality aluminum water bottles instead of plastic bottles. We can use stainless steel bottles, and we can use glass bottle instead of plastic bottles for babies. And we can use cast iron or stainless steel instead of non-stick cookware. And we can use organic and natural cosmetics which are devoid of endocrine disrupting chemicals. And of course, eating healthy, exercising, getting quality sleep, and avoiding stress are still the most important factors when it comes to your health. So only you can decide whether going through extreme lengths to avoid chemicals is worth the inconvenience and the extra cost. No, now you can always relate this lecture to COVID-19 situation. How we can relate this endocrine disruptor chemicals to our COVID-19 situation. Now, as we all know, novel human coronavirus, that is severe acute respiratory syndrome, coronavirus 2, that is SARS-CoV-2, had emerged in Wuhan, China in late 2019, which causes the COVID-19, and it had spread globally, causing unprecedented deaths. Now, a study in 2003 in China had shown a positive association between air pollution and SARS case fatality. So, SARS patients from regions with high air pollution index, that is high API, were likely to die. They were more likely to die from SARS compared to those from regions with low API. Now, obesogens or endocrine disruptor chemicals causes obesity which leads to diabetes, insulin resistance, cardiovascular disease, fatty liver disease, cancer, and various metabolic disorders. Now, as we all know, individuals having various comorbidities are more prone to severe illness from COVID-19. So individuals who are more exposed to environmental pollutants are more susceptible to coronavirus infection. Now, these are the guidelines given by CDC. We all know that people with chronic lung disease or moderate to severe asthma, they are more prone to COVID-19. People who have serious heart conditions and people with severe obesity, that is body mass index of 40 or higher, they are also more susceptible to these COVID-19 disease. Now, and people with diabetes, people with chronic kidney disease, and people with liver disease. So these endocrine disruptor chemicals or obesogens, basically they are causing obesity, and obesity is responsible for causing all these comorbidities, that is diabetes, chronic kidney disease, liver disease, and heart disease. So obesity leads to so many related metabolic disorders, and which are people who are at the higher risk for this COVID-19 have all these comorbidities. So we can link our obesogens with the COVID-19 situation. No, a link between air pollution and COVID-19 deaths. Now, as we all know, that is countries all over the world have implemented some sort of lockdown to slow the infection of coronavirus. Now, lockdown due to COVID-19 has drastic effects on social and economic fronts. But however, this also has some positive effect on natural environment. Recent data released by NASA and ESA, that is European Space Agency, show that pollution in some of the epicenter of COVID-19, Wuhan, Italy, Spain, and USA, etc., has reduced up to 30%. Now, as per European Public Health Alliance, air pollution weakens the immune system, affecting the body's ability to fight infection. Now, a 2003 study found that SARS patients were, as I told you before, that SARS patients were 84% more likely to die if they lived in areas of high pollution, that is high API. Now, new research indicates long-term exposure to air pollution can it could lead to higher COVID-19 fatalities. Some believe that coronavirus on air pollution particulates, they can help the virus spread. And it was also noted that 78% of the deaths in Italy and Spain occurred in the regions having the highest NO2 concentrations. Now, NO2 is highly reactive pollutant and it is emitted from combustion of fossil fuels. Traffic pollution is the major source of NO2 emission. Lockdown reduced transport activities, so NO2 emission reduced significantly due to this COVID-19 situation. So now particulate levels in New Delhi and Seoul, they have also plunged 60% and 54% respectively. And new research published in the journals, that is Science of Total Environment, has found that long-term exposure to air pollution may be one of the most important contributors to fatality caused by COVID-19 virus around the world. So based on the existing studies, there is a very strong correlation between levels of NO2 and deaths associated with COVID-19. Now, as per Harvard study, an increase of just 1 microgram per cubic meter of particulate matter, that is 2.5, correspond to a 15% increase in COVID-19. It is known that certain parts of New York have annual PM, that is 2.5 levels, consistently above the safe threshold. And researchers suggest that this could have played a part in the scale of New York State's coronavirus outbreak, with deaths by far the highest of any state. The evidence we have is pretty clear that people who have been living in places that are more polluted over time, that they are more likely to die from coronavirus, say Aaron Burstein, the director of Center for Climate, Health and Global Environment uh, at Harvard University. So we can prove that there is a link between air pollution and COVID-19 deaths. And now, what are the future directions? Now, today, with the advent of modern food production and distribution practices, access to abundant and palatable and nutritionally dense foods has become the norm of a large fraction of the world population. Now, the mantra of eat less and exercise more is the appropriate response to this altered lifestyle, but it is difficult to implement in our modern societies. But you know, human societies, they have also changed in other important ways, like our exposure to chemical pollutants today is vastly expanded through industrial process that generates metabolically stable chemicals and that our body is not equipped to handle. So like we have to take certain steps to prevent the environmental pollutions. Now, here we see that although the environmental obesogen field is just 15 years old, it is becoming clear that chemical exposures may be important contributors to the obesity pandemic. Many advances have been made into potential mechanism underlying obesogen action and how obesogen exposure may predispose humans and animals to obesity. Now, many of the recent discoveries regarding the mechanism of obesogen action are still in the early stages and require more research to determine their significance in obesity susceptibility. Now, understanding how obesogen act will pave the way for identifying new obesogens that may act through similar mechanisms and what is needed for EDC in general and obesogens in particular are better screening systems that can identify likely candidates for in-depth screening. Now, EP has distinct program for listing EDC. About 52 chemicals published on the EPA website were shown to have endocrine activity. So, a continued improvement in global legislation and adoption of practices such as REACH will see a move to safer alternatives, enabling better green and sustainable chemistry. Now, precautionary principle is the best option where we take steps to eliminate obesogens in our diets, personal care products as much as possible, and we can use organic foods, personal care products which are lacking endocrine disrupting chemicals, and we can also reduce single-use plastics. And we have many buying choices in the markets. We have organic foods, so we can switch over to uh, those food choices, and we can reduce our exposure to the environmental pollutants. Yes, tell me.
मैम इसमें Q&A बॉक्स है, उसमें क्वेरी है endocrine disrupting chemicals hormones act as obesogens by the mechanism.
How to cope with? I have given so many these things now. How we can prevent from these chemicals, like we take steps to eliminate obesogen in our diets, personal care products as much as possible. We can use organic foods, we can use personal care products which are devoid of EDCs, and we can use stainless steel bottles instead of plastic bottles, and we can use cast iron utensils instead of that is what do we say, non-stick cookware. So these are all the precautionary measures we can take to avoid the common obesogens which we encounter in day-to-day life.
One question is there.
Yes, yes.
How spike proteins of COVID-19 attacks our internal mass of the body, and how it goes into the receptor and affect our lung cells?
So actually, I'm a chemist, but I will try to answer his questions. So what it will when it binds to receptors, it increases that is immunological response, as a result of which cytokines are secreted. And we say that if lots number of immunological response, that is cytokines, are secreted, then it causes death also. So that is the immunological response, and it binds with the protein coat which is present on the coronavirus, and it initiates the response in the lung also. So it initiates immunological response, as a result of which cytokine are liberated, and if they are secreted in excess, then it is harmful to the body because they won't recognize the normal or the coronavirus affected cells.
Okay, okay. So we have lots of feedback related to this session. Thank you, Nighat ma'am, for your precious
Thanks
time.
for inviting me. Thank you so much, Dr. Sama, and all faculty members.
Yes, ma'am. On behalf of Poornima Institute of Engineering and Technology, from the management of Poornima Institute of Engineering and Technology, from Dr. Dinesh Goyal, from Dr. Gautam Singh, and the entire team of this event, I would like to thanks Dr. Nighat ma'am for his precious time and valuable information, very informative information given to our participants. We will definitely try to connect with you in future also, so that we can take experience, from your knowledge also. Thank you very much, ma'am, for giving your time and connect with us, ma'am.
It was thoroughly my pleasure, Sama.
Thank you, ma'am.